past 5 years, I've gotten the pleasure of working as a beekeeper here in the San Luis Valley. So when Dr. Stephenson approached me about doing research with honeybees, you could say that I was more than thrilled.
Now, when I was approached about working with the honeybees, as well as Dr. Stephenson was, we were both excited, but also a tad bit skeptical, unlike Russell, as you can see in this picture.
Hi, I'm Russell Geminden.
And I'm Garrett Visser, and today we'll be talking about the immune response to lipid polysaccharide challenge in two subspecies of honeybees, Apis mellifera. Now to begin, starting in the winter of 2006, a mysterious worldwide phenomenon known as colony collapse disorder, otherwise known as CCD, was first noticed. It still exists today, and as of right now, there are no known causes for these collapses, as well as there are no preemptive symptoms that we can look at as an onset to the collapse in the colony. Now, what is colony collapse disorder? It is described as the total loss of adult honeybees in a colony, aside from the queen and a few juveniles, thus leaving the colony looking like this on the right. So between the years 2007 and 2008, the U.S. experienced a loss of about 35% of their colonies, or about 1 million colonies. Now, with some basic math and a rough estimate, that's about 50 billion bees lost in just that year alone, when you take into the fact that at apiaries, a typical colony will have between 50 and 75,000 bees in it. Now, this figure here shows the number of U.S. honeybee colonies on the y-axis when compared to the year on the x-axis. Now, shown here by this blue line, starting in 2006 is when colony collapse disorder was first noticed, by this sharp decline in number of colonies. But if you look towards the end of this figure, the line shows an upward trend. It's a positive thing for us to look at, thinking that colonies are being replenished in a way, but there is still a lot that is not understood about colony collapse disorder and how it affects our global economy and agriculture worldwide.
So with CCD, like Garrett had mentioned, we're really not sure what causes it, but we do have some suspects that are under study right now. One of the major suspects is Varroa destructor, which is a mite that you can see pictured here on the back of the bee. It actually drinks the bee's blood or hemolymph and can be a vector for diseases such as bacteria and viruses. Other pathogens like fungi, shown here is nosema, which is a parasitic fungus that lines the intestinal cells within the honeybees and actually prevents digestion of food, which leads to starvation and a weakened state. Other pathogens include viruses. As pictured here is the Israeli acute bee paralysis virus, which is being studied now and might be one of the viruses that Varroa destructor spreads. There are also many more viruses that we don't know much about, and probably more that are yet to be discovered. Finally, another thing that we would take into consideration as a possible cause for CCD is human disturbance. This includes things just as disturbances as well as pollution and the use of pesticides, as pictured here in almond orchards in California. So why should we care about the loss of honeybees? Most people are familiar with honeybees with their production of honey. So you might think, well, honey's great and all, but if we lose it, will I really miss it that much? But they also provide a lot of other different things that we use and other people use on a daily basis in consumer society, such as pollen pictured here in a frame, beeswax, which is used not only in the industrial realm but also at home, and royal jelly, which is used in beauty products and all sorts of other things. But most importantly, what bees contribute to humans is pollination. Pollination is important because it makes up approximately 1/3 of what we put on our dinner plate at night. In this infographic here, you can see a breakdown of common crops in the United States that are pollinated by honeybees. In the orange, you can see the percentage that the honeybee pollinates as well as other pollinators. If you notice, almonds are 100% pollinated by honeybees, so no other native pollinators that we know of or the use of bumblebees and other crops can really be used to get that. And when you think about that, you might say, well, I can live without almonds, but there's a lot of uses for almonds just besides eating them as the fruit or the nut. They're used with oils, their oils are used, there's almond milk and other things. Runners up are apples and blueberries at 90%. Here you can see the breakdown in billions of dollars that were made in these crops in 2006. So as our society expands and population increases, these figures will also increase.
Now, speaking about billions of dollars, honeybees typically provide annually about $215 billion to our global economy in just their agricultural uses. Now, this figure here, this picture shows what $1 billion looks like. It's 1,000,000 $100 bills. Now imagine seeing 215 of these and having them just disappear from our global economy. And imagine the impact that would have, not just monetarily, but as Russell discussed, in the resources and food that we use almost every day. So in our test, we decided to test between two different subspecies of European honeybee. First was the Italian honeybee, or Apis mellifera, and then we compared that with the Africanized honeybee, otherwise known as feral, Apis mellifera scutellata. Now the Italian honeybee is a very common honeybee used in beekeeping worldwide due to its more docile nature. It's not super aggressive, and it has very good honey production. And we compared it with the Africanized honeybee. It's a little more aggressive in nature. It's not the killer bees that everyone thinks that they are. They're actually a hybrid of the Italian and those killer bees. So they're not as docile, but they're very good at producing honey, comparable to the Italians. Now why we decided to compare these two is that the Africanized are suggested to be much hardier and more resistant to disease compared to the Italians. We wanted to see maybe why that is on the immune level.
So to do this, we used a common, in this graphic here, the common pathways used by invertebrates, such as insects, including honeybees, to try and get an idea or an early test for ways that bees might upregulate their immune system different from one another. We focused on prophenol oxidase and the products that are led past this, such as melanin. Melanin leads to encapsulation, which is one of the main pathways for fighting a disease. Encapsulation basically covers viral pathogens as well as bacterial pathogens that doesn't allow them to infect the host. This is usually a later stage of an immune response, so bees typically and other invertebrates usually use reactive oxygen species pathway. Reactive oxygen species pathway uses reactive oxygen, which damages parts of cells as well as genetic material so that it will kill off bacteria and viral pathogens. Now, with reactive oxygen, it is also dangerous to the host. So the host actually will make antioxidant proteins that deactivate reactive oxygen so that doesn't harm the host, but does kill the bacteria and viruses. This is a first line of defense and one of the pathways that we use to study. So our objectives in this research were to identify if there really is a difference in upregulation of the immune system between the two honeybee subspecies. Another objective is to see if this changes over time when incubated with a stimulus that would make the immune system upregulate. To upregulate this immune system, we use something known as lipopolysaccharides, or LPS. Lipopolysaccharides are fatty sugars that are found on the extracellular part of gram-negative bacteria, usually associated with infections. So an example of gram-negative bacteria that has lipopolysaccharides on the outside is E. coli. In fact, in our study, we got laboratory purified LPS that we would then inoculate the bees with so that it didn't have a pathogen associated with it. So the bees weren't really getting sick. We were just making them think that they were sick so that the immune system would upregulate so then we could conduct our tests. We did this by incubating 3 sets of bees. One control, which was instantaneous. Right when we inoculated it with the LPS solution, we extracted and did testing, and then we did a 2-hour incubation and a 4-hour incubation.
Now, to begin our testing, we first had to obtain our samples. So what we did was we worked with local farmers, went out into the field to collect our honeybees. What's seen here is what's known as a queen excluder that we're putting on top of the colonies. It's basically a metal screen that when we encourage the honeybees to move up through their brood area into what's known as a superficial structure, or this box that we keep our honeybees in, the queen is not small enough to fit through that, so she's actually not included in our sample. So she stays so that the rest of the colony can continue to thrive and work and live. So seen here in this video is actually how we did that. Here Russell is using burning burlap sack and blowing the smoke through the bottom of the colony, which encourages the bees to move upwards through their brood area, through the queen excluder and into our superficial structure. So here's the bees actually there with the smoke. In this video, we have our honeybee filled super, and we are shaking it above this here funnel so that we get a random sample of honeybees that fall into our homemade container that we used transport our bees in. What I'm spraying here is a sucrose water solution onto the bees. It inhibits their flight, it's kind of sticky, and it keeps them more docile while in this container as we transport them and get ready for the assays.
So when we bring this container back to the lab, we allowed it to stay in an incubator at approximately body temperature, so 37 degrees Celsius, overnight. And what this does is allow the bees to become really hungry. So essentially we do a starvation technique so that the next morning when we inoculate the bees with the LPS sugar water solution I was talking about, they take up the full aliquot so that we can standardize it between all the bees to make sure that they have equal parts of the LPS solution that we mixed up so that everything is standard and that doesn't leave any room for error. Bees were fed using a micropipette seen here. With a 1 microliter aliquot of the LPS solution, and this can actually be shown feeding the bees in this video here. So you'll notice at the very end the droplet and the bee using its tongue to essentially lick up the LPS. So from here, we would then incubate the bees for the time that I was discussing before. So bees at the control were fed the substance and then immediately having their hemolymph extracted. Hemolymph was extracted by removing the abdomen from the thoracic cavity and head, where then it was dorsoventrally compressed until a bead of hemolymph could be collected. Any bees that didn't provide enough hemolymph, which was about 2 microliters, were discarded and not used in the test. So we made sure that we had equal amounts of hemolymph for every sample. We went through the other incubations for a 2-hour time period and a 4-hour time period before collecting hemolymph from those bee samples as well. These samples were buffered and placed in a buffered solution with glass beads, where they were then homogenized or vigorously shaken to break up any cell components and make sure that all the proteins we wanted to test were accessible to our assays. These were kept at -80 degrees Celsius until we began colorimetric immunoassay tests. Color immunoassay tests are used to basically see the percent activity or total concentration of the immune pathways I discussed before. Over time, the assays will change color, which can be read by the spectrophotometer here in the 96-well plate you've seen here. Down here, you can actually see a slight color change in one of the wells compared to here. It's a light yellow, orangish. And that will actually be read by the machine so that we can actually see total protein concentration or get an idea of activity of these proteins in question. The tests that we ran for this study were protein concentration, total protein concentration using Red 660, which just looked at all the proteins that might be upregulated or downregulated over time. We specified in one of the pathways I discussed before, the reactive oxygen pathway, using that antioxidant protein that protects the bees known as superoxide dismutase.
So our first set of data that we really gathered, and we weren't exactly even looking for at the beginning, but we did notice, was survival. So on our y-axis, we have our percent survive between the 2 subspecies of honeybees compared with our 3 different time points on the x-axis. Now for our control, which was 0 hours, no bees died, as you would think, as we're not actually infecting them. It's just like a lipopolysaccharide as a challenge, so they're not actually becoming infected. When you're looking at our 2-hour time point, only 1 bee died from each subspecies, so there's still no main difference, as you can see by the 2 A's, suggesting that they're basically the same. Now we're looking at our 4-hour time point. Despite it not being statistically significant, it was notable that there was a decrease in percent survive in both subspecies, and especially in the Italians. Our second set of data that we had was our protein data, or using our Red 660 assay, which looked at protein concentration on the y-axis, and our 3 different time points again on the x-axis. Now with our control, the Italians had much higher protein concentration compared to the ferals, which shows that they have higher basal immunity, or they're always kind of ready for an infection to come whenever it happens to come. Now when we look at our 2-hour time point, the Italians stayed about the same at about 0.5, but the ferals really ramped up their protein concentration all the way to the point that there was no significant difference as seen by the 2 A's. Now we're looking at our 4-hour time point. This is where it gets more interesting. So with the 2 B's on each of those 2 bars, that indicates the significance between the subspecies at this time point, and that there was not any real statistical significance here. But with the A compared to the B on the red, that's when we coupled subspecies with time. and that was statistically significant with a p-value of 0.05. Now, this is intriguing for us because it shows that between the two subspecies, they change how they regulate their proteins over time, which is what we were actually looking for. And our third set of data here is our superoxide dismutase test, where we looked at percent SOD activity on the y-axis, and again our 3 different time points on the x-axis. So for each of these different time points, there was no statistical significance and difference between the subspecies at these different time points in their percent SOD activity. But if you were to look at all of our samples put together and compare that with just time, there is an upward trend here, and there is a significance in that data of p-value that is less than 0.001, or that's very significant. So what does this all mean? In essence, with the survival, there is a survival component, because if you think about it in the biological sense in real life, when a honeybee is infected, it doesn't just stop at 4 hours. It continues to be infected until it either fights it off or the honeybee is killed. So there is a relevant survival aspect to this challenge and what we're looking at. With our protein regulation, it is shown by our data that the 2 different subspecies do differentiate in how they regulate their proteins, as the Italians had a lot at the beginning, but the ferals, or the Africanized, really began to ramp theirs up over time. So it shows differences in how they fight off this infection. For our SOD activity, it is proportional in that there was no real difference between the 2 subspecies at any of the time points, but when looking at just time, they do increase it over time, but at a more slow and steady rate, leading us to believe that maybe they're using a different pathway to fight off this lipopolysaccharide challenge.
So we actually have tested another pathway, a third pathway known as the prophenol oxidase pathway I mentioned before with melanin synthesis. We couldn't actually get the data in for this talk, and so we don't really have any conclusions for it, but we do know that there's a lot left to be done and studied. Other studies that we can do for this might include things like genetic studies, looking at how proteins are upregulated and downregulated on a genetic level, rather than a classical technique such as looking at proteins first. So we'd like to acknowledge Brent Edelin, a local beekeeper in town, for showing us the bees and lending us the equipment and his knowledge and expertise in the field. We'd also like to thank Dr. Matthew Stephenson for allowing us to conduct this research underneath him and showing us how to do this the proper way and giving us this opportunity.
And then we'd like to thank Adams State as well as the Department of Education for allowing us to have funding and the resources to be able to run these tests and experiments for the last few years.
So here are references. We did have a really cool one that was on bumblebee workers, and that helped quite a bit. And if you're ever interested in any of that kind of stuff, it would be a good read. Are there any questions?
So I'm curious, you mentioned in recent history there's been sort of an uptick in terms of bee populations in the United States, and it's still uncertain exactly why that's the case. Are there any theories at the moment for what might explain that?
Coming from a beekeeper's standpoint, I think the concern of CCD really, it shocked a lot of us because of how drastic it happened, and I think a lot of practice within beekeeping changed. So people started to become more conscious about wearing rubber gloves instead of using reusable gloves that would spread disease in colonies, being conscious of how to treat those colonies the correct way with antibiotics, getting more educated and asking scientists for help with this. So that might be partially due to it, but to be honest, I don't think we're really sure, and I don't know for sure.
And one thing I read about that actually is that because of colony collapse disorder happening, it's intrigued a lot of people to maybe get into beekeeping to increase the number of colonies. So what I've read is that a lot of people have been going out and actually buying queens and then buying colonies and trying to become beekeepers to be able to increase the number of colonies and bees we actually have.
When you guys were figuring out your methods, was there anything you tried that didn't work?
Well,
not right away, not that we had noticed.
no, we had done some assays before that we had some trouble with, and so we were trying to optimize things that we learned from our last research. So for the most part, this actually went fairly smoothly. We do have a lot of questions still, and there's a lot to be done.
Okay, but from your previous research, you were able to kind of take the lessons learned from that. and
Right.
apply this to
So like last year when we were doing other research, we ran very similar tests. So we figured out the correct protocol for that. and was able to implement. We were able to implement that in this research, so it ran a lot more smoothly. Yeah.
And then with the people question, like how are the humans affecting
Mhm.
what's happening here? How do you study for that? Because I mean, the environment, you can't go back in time to a pre-human environment and see what's happening.
Well, basically, you can look at what pollination is and how it affects the amount of food that we eat every day. And you can base that and look at people's diets and see if they're not getting these nutrients from these foods, how they're going to be affected in that way. So you can see how they'll be affected from the honeybees being gone by their pollination through the food that we eat.
Right, just compare with the colonies in the past, you know, like.
Colony health. So a lot of research, you know, back in the day, because the bees were doing so well, I don't think a lot of research was really being conducted. So most of it is kind of like a first-hand experience, like talking with other beekeepers that have been in this field for at least 40 years to before. If you look historically how they talk about the bees, as you know, like writing more of a historical sense than actually a scientific sense, I think is a good way to get kind of a feel for how much it's changed. Just talking through some families that have done it for as long as they've been in the United States, so almost 100 years, they have noticed and documented themselves how much of a change it's had, and a lot of that has to do with urbanization, how we keep the bees because of how people have distributed themselves and made it less accessible for beekeepers to place their colonies where they need to be. But as far as scientific research, I'm not sure.
There's not much on that. So. Cool.